we're going to be looking at developmental biology. I think that this is such an interesting field that is taking off right now. 2018, we had a really cool, actually Science Mag named it the breakthrough of the year, because it's just has gotten to this point where I think our imaging capabilities, the ability to make mutations with fluorescent markers, meaning that we actually have fluorescent proteins that could be bound to proteins and follow that throughout the cell, and then also specifically for developmental biology, kind of like seeing how that organism grows up for the first time. So do you have any thoughts about that, Miller, before we watch the video?
Yeah, man. I mean, if you think about kind of some of the breakthroughs that we've had, now Dr. Chen, who's the guy who developed the GFP protein.
Mm-hmm. UCSD.
Like, what's that?
That UCSD, right? You got the Nobel?
Yeah, UCSD. He passed away recently. But, you know, just a simple breakthrough where you can, like you said, follow things around the cell and just really visualize what is happening. And, you know, before that, we were only able to kind of just imagine, and then to see things happen really change the way we view cells and how they function and the things that they can do. And so I think this paper that you speak of is just another example of the use of that technology.
Yeah, because like in the last, 10, 15 years, we've had high throughput sequencing abilities kind of being developed too. So I think it's kind of like a merger of two worlds, right? Where you have things like FISH, fluorescence in situ hybridization, where you're actually able to see specific genetic DNA combinations be highlighted inside of the cell, like whether or not that actually has that sequence within its cell. And then you have kind of coming out of the Church lab too, using CRISPR on top of that to target specific DNA sequences within the cell, and then do real-time sequencing within the cell. I mean, that's just crazy to me.
Yeah, I mean, it's kind of really fun to see, you know, all these technologies that we talk about, and, you know, they're developed in basic academic labs, which, you know, sometimes people don't understand. You know, the reason why we as a country spend so much more money on research and development than in other countries is because these basic discoveries just lead to things that actually have a drastic impact on our everyday life. And, you know, I think sequencing is just another example of that. You know, you see a company like Illumina who got, you know, 18 small business grants when they first started their company. And without that, they wouldn't be the company that they kind of are today, that is just, you know, changing the way that we think about science and disease.
Mm-hmm. Yeah, definitely. I mean, there's no way that, think of all the grants and applications that Doudna was writing for all those years talking about antibacterial resistance. You know, it's like, oh, no, this is important. This is important. No way that she's thinking, you know, one day that she's going to make this genome editing tool. But I think it's a great example of, research versus engineering, right? Where research can kind of like help with that.
Yeah, for sure.
All right, let's watch this video.
How do many kinds of cells arise from a single cell during development? Single-cell sequencing technologies and new computational tools are providing the most detailed picture yet of this process. In several types of studies, embryos of different stages were separated into individual cells, and the cells are sequestered away in droplets of oil. What kind of cell am I looking at here? To find out, the researchers sequenced each cell's messenger RNA, which reflects active genes. Each cell type has a profile, a set of active genes that define its identity as a muscle cell or a type of brain cell. How do I track a cell through time? In one line of experiments, researchers analyzed some 92,000 zebrafish cells and gathered data from 7 different embryo stages. To help tie the cells to a time point, they added genetic barcode DNA to the cytoplasm of some of the single-cell embryos. These are many tiny pieces of unique DNA that will be incorporated over time into the genomes of daughter cells. By the end of the experiment, the combinations of barcodes reveal how the cells are related. Thus, they could trace each zebrafish cell's fate through time to see how a single cell gave rise to a variety of specialized cells.
Oh, wow, that's incredible. That was so cool.
That was crazy.
You really see it go like in real time there. I mean, just all those embryos of the zebrafish at one time. You can just see them all develop like that. That's just, and then sort too at the same time, knowing what's happening inside that. Each of those cells. I mean, it's just incredible. And it's at the population level, right? It's not just at the organism level. We're like just a collection of single cells, if you think about it. And you can really like see what's happening inside each of one of those cells as it develops. I mean, it's just so cool.
Yeah, man. What's crazy is, I think a lot of people sometimes ask, well, why are we studying this zebrafish here? And just the thought that, developmental biology and the things that we can learn to, can hopefully lead to one day, our ability to use stem cells to grow an organ, for example.
Yeah.
To really save someone's life. And just watching this kind of just is amazing to see kind of real time.
Mm-hmm. Definitely.
Such as heart, nerves, and skin. What can this tell us? This approach is already.
So we have a question here in chat. Frank Fields, that turns into a zebra.
Zebrafish. And yeah, it's a zebrafish. Yes. It's a small little fish that's yeah, you can actually buy them at pet stores. They're like everywhere. What's crazy is that I was actually at PetSmart, and they actually have like, I forget what they're called. I think it's like Dayglow or something they call it. There's actually a patented name. They actually they genetically modify
Dayglow?
Yeah, they put in these markers to actually make them glow over time, and you can actually, they glow. The fish literally glow, and you have like UV lights that you can put in the aquarium tanks, and you can actually see them glow different colors. Pretty cool.
Is that safe for the fish to have day glow inside of there?
Well, it's just a fluorescent protein, right? So it's not like the actual, is Dayglow something else? I don't know if that maybe that's... It's just a fluorescent protein. So theoretically, it's not going to be able to compete as well against in the wild versus a zebrafish that didn't have to make that protein, right? That's like expending resources, but it should be fine, yeah.
Perfect. And then I think we'll go ahead and field Joe Ostrin's question, where do babies come from, on a later podcast.
Biologists have thought that once the cell starts down a path,
we'll get to that. We'll fill you in, but there's a lot of good books out there on that.
Yeah, that's the next one.
Yeah.
Researchers observed
crazy
some
story.
zebrafish cells.
I came home to a random stork walking across my yard.
Uh-oh. Did you get a baby?
My neighbors did.
Different.
Oh, perfect.
Congrats to those neighbors.
Yeah.
Types, as indicated by the change in what genes were active, after they should have been well committed to a certain fate. In frogs, gene activity showed that even when an embryo looks like an undifferentiated blob, cells are already starting down the paths to their eventual identities. With these tools, researchers can also see how development differs from one animal to the next, or even learn a recipe for coaxing different cell types from stem cells.
Wow, that's so cool. So the reason why I bring this up, and unfortunately we have 5 minutes left in our Zoom meeting. I did not set up the... my school does not subscribe to Zoom Prime, apparently. The cool thing that I wanted to talk about this was that basically labs are trying to grow brain cells through cell culture to see the impacts of COVID-19 on their brains.
Wow.
So it's kind of
it seems like there'd be other implications for that too.
Yeah, exactly. So that's kind of one of the problems that they're talking about here is like, can these cells become conscious, and what's the kind of ethical implication there too? So yeah, I think it's really interesting. Where is that? I can't find that article. Let's see.
I mean, I think that's a very interesting question. Where does consciousness kind of begin? And is just one type of brain cell sufficient to do it, or do you need this, the whole nervous system that kind of gives those brain cells the experiences of the world?
Yeah, here we go.
Really say that.
Yeah, right.
I mean, I think like as humans, we intuitively kind of put ourselves at the top of a pyramid of consciousness, but over how many centuries now of research has that been wrong? I mean, after every step we have defined this line, and we have been wrong at every single stage. I think that, and I have like whatever the question is though, whatever the answer to that question is, whether these brain cells are conscious, I still think that there's some validity to studying COVID-19 in brain cells because, I mean, primate research is banned, right? So and I mean, rat brains is kind of tentative at best. So I really think that brain cells in cell culture would probably be the best way to do it.
Wow, that's interesting.
Hmm.
And yes, and if you're not aware, there's been several studies now that have had shown long-term neurological issues with COVID-19. COVID-19 obviously being the disease caused by SARS-CoV-2, and the long-term neurodegenerative disease that comes with it. And I mean, we've had like a lot of anecdotal evidence about like people being foggy and feeling like they've had dementia for months.
Yeah.
It's really pervasive right now.
Yeah.
And we don't know if that's going to be lifelong or what, or a temporary condition.
Exactly.
Yeah. And it's kind of an interesting thought because, the blood-brain barrier is really built to keep things like a virus out. So you'd almost have to believe that the virus has some way of disrupting that blood-brain barrier to gain access to the brain.
Yeah,
right.
Exactly.
Are there angiotensin receptors there? Isn't that what they need?
What are those? What are angios?
Well, I'm asking the scientist here.
I don't know.
I am your non-science voice.
You're talking about the COVID-19 receptor that
Yes.
targeting development, for example.
Okay. ACE2.
Yes.
Yeah.
The ACE2
Gotcha.
receptors. They bind to those receptors. So maybe that's how they're crossing.
Those receptors are definitely on brain cells, and so there's a reason why you would study it there. But I guess then it begs the question. I don't know about the blood-brain barrier and anyone finding that receptor on the blood-brain barrier.
Right.
Might be some random inflammation that's being caused, which is kind of interesting too. We've been talking to a couple groups even in our world about develop some therapies, and one of the things that they were talking to us about was, aside from just the respiratory issues, just a lot of the organ failures that you see in people that have COVID-19, and so there's just so much more about the inflammatory side of this disease that's still a little unknown.
Right.
Interesting. Well, I think we're gonna try to get a premium Zoom link set up real quick, but for right now, while we do that, all right. Well, that's good. Yeah. All right. Well, that happened. All right. Well, I mean, I think developmental biology in general is just going to be such an interesting field. We're learning so much more at the organism level. I think that's the biggest thing, is that we're finding now about things that are happening, across the entire organism, and it's not just cell by cell, micro by micro, which is kind of a field or a way that we used to look at things through microbes, right? That microbes were one of the reasons why such a powerful tool, because you could see so much what was going on. It wasn't just a single cell, a single organism that you're looking at. You're looking at millions of cells. So it was kind of like a black box, kind of like what's going on with these huge organisms that are a collection of cells, for example, the human body. And developmental biology with imaging techniques, and imaging is only getting better, right? I mean, the different types of imaging, the different wavelengths that we can see, we're only going to be able to see more, sequencing in real time. And because if you think about it, in order to sequence an organism, we've had to pull the DNA out. We had to pull the DNA out of the organism in order to actually look at the DNA sequence, but not anymore. We can actually sequence the whole thing at the same time and look at things. So, one second, sorry. So cool. I think it really is a very, very cool time for science. Thank you so much for watching. If you like this content, please subscribe to be notified of our latest live streams where we have scientific debates, informative conversations about issues, and follow the latest advances in science. If you want to join a thoughtful, non-toxic community where we have good discussions about science and other issues, please join the Discord. The link's in the description. Thanks again.